yesterday's recording because it was so bad, because I didn't have pictures and I didn't have the capability to draw like I thought I was going to. So that one I'm probably not going to load. I would remind you though, if you're tuning in today and you weren't with us yesterday, that the bacteria that we covered specifically that cause human diseases, we talked about Clostridium tetani, botulinum, and difficile, and we also discussed Listeria monocytogenes. So you'll want to look into those a little bit and see their general characteristics and some of their, what makes them a particular pathogen. All right, so back to chapter 28. Again, I'm not going to go through all of the general characteristics. I would just keep reminding you that protists are eukaryotes. And I know that's difficult for many of you because prokaryotes and protists both begin with pro, and they're both small forms of life, and they kind of all blur together, especially here at the end. But you have to remember, prokaryotes are bacteria, so domain archaea and domain bacteria, and protists are all eukaryotes. So I was going to sort of just share this. I didn't pick out particular PowerPoints. These are just all the pictures from your book. This is probably one of the most important pictures in the book because it is showing us sort of the evolutionary history or the phylogeny as we know it today. This is changing all the time, and probably in the next edition of Campbell, I would suspect a few nudges, a few minor changes here and there. So the protists have generally been divided into 4 major groups here. Excavata, STAR, again, that's for abbreviation for these 3 major subgroups, the Stromatophiles, the alveolates, and the rhizarians. The Archaeoplastida, this would include the ancestors to green plants. You can see plants showing up here. Notice they are not highlighted in yellow because they are not protists. And then Uniconta, which of course includes many of the molds, the unicellular molds in the amoebozoans here, and the pisthecons, that's going to include the fungus, the choanoflagellates, which are the ancestors of sponges, and then the rest of the animals, which we would fall under this category here. So your textbook is going to repeat some of this beginning stuff here in the first chapter about plants, which is chapter 29, and I would encourage you to take a look at that. The similarities and differences between land plants and particularly the charophytes and chlorophytes. And then again, at the beginning of chapter, I believe it's 32, they're going to revisit, well, of course, during the fungus chapter, which is 31 maybe, they're going to talk about a little bit more about the nuclearids, and then the beginning of the animal chapter, which I know it's pretty sure is 32, they're going to revisit the choanoflagellates. So some of these characteristics and things that I'm going to mention, you may not find them in chapter 28 per se, but if you dig a little bit and look forward to the first plant chapter as well as the fungus and the first animal chapter, you would find the connections to the rest of the protists. Too heavy to keep on my legs. So, sorry about that. All right, so let's look at, we've got these 4 supergroups here, and let's start with Excavata, because that's where your book starts. And this is a representative here. This is Giardia intestinalis, or looks very similar to Giardia lamblia, which is a parasite of humans as well. And you'll notice that these belong to the diplomonads, which is one of the subgroups of Excavata. You'll notice that they have multiple flagella, right? So these are single cells. They're, some of them are free living and some are parasites, and they're of course aquatic. They're going to use their flagella for motility. Another important group, I'm going to keep coming back to this original slide here, or this breakout. So those are, the Giardia are diplomonads. The parabasalids, those are going to include Trichomonas, which is another parasite of humans, can cause disease. Both of these groups, they have what is known as a mytosome. And a mitosome is similar to a mitochondria, but it's almost like a degenerative mitochondria. It's believed that these organisms originally had mitochondria and then they have lost them or modified them. I believe all of the parabosolids are parasites. Probably a picture of, there it is. So here's the undulating membrane. That's one of the stellar characteristics of Trichomonas. And so in addition to flagella, they use this undulating membrane that kind of moves in a wave in order to infect and swim. And then I think one of the more interesting groups out of the excavata are the euglenozoids. And these are going to include the kinetoplastids, which is trypanosomes, and the euglenids. Did I show you a picture of a trypanosome there instead of, no, it was Trichomonas. Okay. So let's take a look at euglena, because this is commonly found in pond water, and you've probably looked at this under a microscope at some time in your life. Single cell, right? One of the interesting things about the euglena is they do have fully formed chloroplasts, which you can see highlighted there. They have both autotrophic, and there are also some euglena that are heterotrophs, meaning they do not have the chloroplast. Most of them are aquatic, or if they're living in soil, it has to be really, really moist soil. They have a unique flagella, which is highlighted here. They have rings of microtubules, and then the rods are made out of this crystalline structure. So you can see this is the flagella in cross section. Looks like a transmission electron micrograph. They do have what we call an eye spot. This is not so much something that can see per se, but it is able to detect light. And so the euglena can swim toward the light or away from the light if it's too bright. You need the light, obviously, for the photosynthesis. I mentioned the chloroplast, and then you can see this, what we call a pellicle, which is almost like a thin rope that's wrapped around in protein bands in this sort of circular pattern around the euglena. It helps give it some structure and rigidity. Most of the time under the microscope, you can only see the long flagella, but they do have two flagella, a long one and a short one. And interesting fun fact about the euglena is that there are euglena that are heterotrophic, and so they don't have the chloroplast, and they are going to have to eat something in order to survive. And if they run into a euglena that has chloroplast and they attack it and digest it, then they actually keep the chloroplast, incorporate them, and become photosynthetic. So it's kind of like a modern day example of endosymbiosis, not just of a whole organism, but of a particular organelle. All right, so we'll go back. Okay, now the SARS group, go back here. All right, so we finish up with euglenozoan. The SARS group includes the stromatophiles, the alveolates, and the rhizarians. And I'm just going to pick out a few that are interesting and or that we have a lot of pictures of. So the stromatophiles, that literally means, I believe, hairy flagella. So you're going to see, we're going to look at some diatoms, and then the golden algae and the brown algae. And the brown algae we're going to spend a longer bit of time on because they have the alternation of generations life cycle, and they're also multicellular. And that's the first time we're going to see multicellularity in this group of protists here. So let's take a look at some diatoms. They look like this. They are normally, their cell walls are made out of silica. When the diatoms die, and the cells themselves die, they leave behind the silica. So if you've heard of diatomaceous earth, which I believe is still used in some aquarium or aquarium applications for filtration, that's what it is. It's basically the shells or the tests left over from diatoms that have died. The protists have died and their shells are left behind. Usually it looks kind of like a shoebox. We don't have a good example of any of those here, in that one of the shells is a little bit larger or fits over it like a lid. Most of them reproduce asexually. They store their energy as laminarin, not starch, which is important. And again, this is just a collection of diatoms. The brown algae, let's see if I can find a picture of that. Oh, here's a picture of the hairy flagella. And here's a super scanning electron micrograph of a diatom. And you can see a little bit more how it's more like a shoebox or a pillbox structure. It has those two parts. Here are some golden algae. There aren't many of these. Most of them, I believe, are like this, either unicellular or multicellular, and they're microscopic. See how tiny they are? So they would be found, I think most of them are actually not freshwater, they're marine. They also store their energy as laminarin, and they usually live in colonies like this. They're not truly multicellular, but they live in colonial groups. And then, like I mentioned, the brown algae. You may have seen these if you go to some tropical places, particularly in the Caribbean. This whole little structure here is tiny. It's maybe, so you can see the representative mollusks next to it. It might only be 3 or 4 inches tall. This is a super large brown algae. And this one is used as a demonstration, again, of the alternation of generations, which that life cycle, remember, we learned about when we talked about meiosis. So these guys are truly multicellular. They have these, which we refer to as the holdfast. They're analogous to the roots. They are not homologous to roots. A stipe, which is analogous to a stem, and then we call these the blades, which again are analogous structures, not homologous structures, to leaves. So most of the photosynthesis is going to be happening up here. These are usually quite long, and you would see them floating with the blades, sort of waving in the water. Again, alternation of generations. So we have a sporophyte that's going to give rise to spores. Via meiosis, they're going to produce 2 different types of gametophytes. Notice the gametophytes look different, but they're multicellular, and then they're going to produce gametes. The male gametophyte is going to produce sperm. The female gametophyte is going to have a little bud on it that the sperm is going to fertilize. We have a zygote, and then that zygote, of course, is going to develop back into a sporophyte. And again, you can go back and review that from the meiosis, I think it was the meiosis chapter. All right, the alveolates. Here's an example of one of those. They are called that because there's a little, these little pockets here near their flagella that are gas-filled spaces. One of an important alveolate would be this dinoflagellate here. It has these 2 flagella that sit in these grooves, so it actually kind of spins. It is often the culprit in a red tide, which you can see here. All right, which these red tides can be driven either by an influx, so a seasonal influx, or an influx of organic material from, for example, fertilizer or untreated sewage. The apicomplexans are also a group of alveolates. The most famous would be the parasite that causes malaria, Plasmodium. You can see it here infecting a person, and it has a really complicated life cycle. And I'm not going to go through it with you, but they're all parasitic, I believe, the apicomplexans. Right. So this is the whole life cycle. Part of the life cycle is completed inside the mosquito, so that when the mosquito bites you, it actually, in its salivary glands, where this happens, the oocyst bursts into sporozoites in the salivary glands of the mosquito. So when the mosquito bites you and sucks some of your blood and leaves some of its own fluids, it's going to inject these sporozoites. They're going to go to your liver and reproduce, and then those gametocytes are going to infect your red blood cells. This is why the fever comes and goes. And then again, another mosquito is picking up the gametes here, and it's going to complete the life cycle in the mosquito. And then last but not least of this group are the ciliates, and we usually use paramecium for our ciliate picture. Here's one that's not as complex. You can see that they're covered in little, what would be like flagella, 9 pairs of microtubules with 2 in the middle, except they're much shorter. They swim very fast. These are all free living, and most of them are freshwater. They have 2 nuclei, which they can actually exchange the micronucleus with another paramecium for increased genetic variability. They attract their food this way. They basically brush food into their gullet here, which then parcels this off into a vesicle for digestion. And then they have these little water contractile vacuoles, which you might remember if you think all the way back to the osmosis chapter, which I think was chapter 7. All right, we have some other examples of the ciliates. Stentor, we usually look at in the laboratory. This is also a ciliate, and the cilia here are around the edge. And sometimes they have this trumpet shape appearance when they're attached, and other times when they're not attached to a substrate, they look more like a paramecium except rounder. And they change shape, but that's how they move, is with their cilia. I tore out. Oh, excuse me, I don't have any pictures of Vorticella, but that's on there because we would have seen Vorticella in lab. Okay, that takes care of this 2 group, the S's and the A's. We'll take a brief look at rhizarians here, particularly the radiolarians and the foraminifera. This, I believe, that's a rhizarian. Let's see, where does that belong? So I guess that's all of these. Oh, rhizarians, right, sorry. Even I get them mixed up. So what you're supposed to be noticing out of this picture is they have these extensions that are made out of either calcium carbonate or, in this case, or silica. And they project outwards. And then again, like the diatoms, there's a test or a shell, if you want to think of it like that. We don't call it a shell, we call it a test. And it's either going to be left over when the protist dies, either made of calcium carbonate or silica, depending on which kind of rhizaria it is, if it's a foraminiferan or if it's a radiolarian. The radiolarians are currently alive. Most of the foraminiferan are fossils, okay? They can be used to date geological rock, and they're also used to find oil. Actually, many of them, certain groups of them fossilized. When people are drilling for oil and looking for oil, they look for certain fossilized foraminiferans in order to be able to tell whether or not it's going to be a good place to drill. And the cercozoans, I think there's just one picture of a cercozoan in your text, and it's way along here. I think this is it. And it has a what we call a chromatophore, so it's not a true chloroplast inside of it. And I think that's the only picture that we have, right? So this is a second case of primary endocytosis or endosymbiosis. All right, now let's go back to our diagram here. So we finished the SAR group, and if we go back to the overall picture, now that brings us to the Archaeoplastida, which are a little bit more interesting, I think, because that's where the land plants originated from or have their closest relatives. So you'll see in Archaeoplastida, we've got the red algae, the chlorophytes, the charophytes, and then plants. Notice the chlorophytes and charophytes are both sort of generally characterized as green algae because they are green, right? And the red algae separate. Notice the red algae is sort of the longest lineage, right? And you might remember from our discussions of endosymbiosis, this is actually a quick diversity of plastids produced by endosymbiosis, meaning red algae as well as the green algae, and ingesting particularly those chlorophylls. But if you notice that the stromatophiles and the alveolates probably actually ingested a red algae, not a green algae. And that has to do with how they store their energy as laminarin, not starch. These guys, the green algae, are going to store as starch. Okay, so let's take a look at some of the red algae first. Scrolling along here. Here's some. So here are just some examples of red algae. My understanding is all the red algae are primarily live in warm water, and they are primarily marine. They're not freshwater. This one you can see is microscopic, or barely, you can barely see it, 8 millimeters. This is larger, right? And of course nori, which is grown to be the wrap for your sushi, that grows much larger than that. This is obviously a little farm of it. It doesn't grow like on strings in the ocean. And these are just bigger pictures of those, right? Characteristics of red algae that are important: chlorophyll A and B. They're multicellular, and they do exhibit alternation of generations, but notice there is no flagellated stage. So that means they do not have flagellated sperm or gametophytes. They just float from one structure to another in order to form zygotes. Here's some pictures of some chlorophytes. So we have zygonema, which is like spirogyra, which you would find in ponds, so freshwater. Here's some ulva, which is a sea lettuce, and a much larger, notice these are both, we would say, multicellular. This is a filament, might even categorize it as colonial, which, let's look at the volvox, which is up here. This is a great example of a green algae, a chlorophyte that is multicellular. It is a giant cell. It looks kind of like a basketball with little tiny cells inside of it, little areas inside of it. And notice each cell has a flagella. See how they're all lined up here? And so when it swims, it spins. These are actually almost like sex organs. These are going to be producing the gametophytes, male and female gametophytes, that are going to be released into the environment to form zygotes and form new volvox. So here's one that is not actively reproducing. Over here you can see it doesn't have as many of those internal structures. Archaeoplastida, and in particular, I would say chlorophyta, or green algae. Okay, so other important features of those green algae: they're storing energy as starch. They have the same accessory pigments as land plants, which means in addition to chlorophyll A and B, they have carotenoids and xanthophylls, just like land plants. Some are unicellular, some multicellular. You've seen that. Some are colonial, and some are truly multicellular, or at least multinucleated filaments, which I guess is the example of the zygonema there. The charophytes, I don't know that I have a picture of a charophyte. This is Chlamydomonas. This is a chlorophyte that undergoes alternation of generations, even though it's a unicellular organism. So that's kind of interesting. And we saw a picture of this way back in chapter 6 as one of our examples of cells with the big nucleus and the chloroplasts and the flagella and the cell wall, similar to a plant, right? And again, they undergo alternation of generations. So they have both, they reproduce sexually and asexually. So for a while, people thought these were the ancestor to land plants, but it turns out that's probably not, right? So if we look here, we can see, actually, I'm going to go back to the original. Let's go back to this picture here. So we're here, red algae, green algae, chlorophytes, charophytes. Now, there's some important features of charophytes that I wanted to point out to you that, again, you're not going to probably find in this chapter, but you would have to dig a little bit into the beginning of the land plant chapter. And I guess I don't really have a picture of a charophyte to put up here, so I'm gonna leave this up. And I'm just gonna list them out. Some characteristics that charophytes share with land plants: they have rings of cellulose synthesizing proteins. So they begin to make what is more commonly like a cell wall made out of cellulose. The structure of their flagellated sperm, because the charophytes do have flagellated sperm, unlike the rhodophytas or the red algae, is similar to plants. And they also form what is a structure called from magoplast, which is the precursor to the cell plate during cell division, during mitosis and meiosis for them. And that's similar to land plants. Unlike land plants, though, the charophytes do not exhibit true alternation of generations, and they also lack, they also show, they lack an embryophyte, which is the defining characteristic of land plants, and they have this protein called sporopollenin, which is to prevent the zygote from drying out. So those are some characteristics they share with land plants, but then there are some that they have different than land plants. So notice their closest living relative, right? And then next closest living relative would be the chlorophytes. And then the last group, the uniconts, is super, super diverse. In this group, we would have, oh, there's my picture of the radiolarian, which I was trying to show you earlier. It's one of the SARS group at the top. All right, back to the uniconts here. So we have amoebozoans, which include the slime molds, both the plasmodium slime molds and the cellular slime molds. This is an example of a plasmodial slime mold. You can see it here. Jasmine, you're going to have to wait till the end because I'm in the middle of lecture. So this is a plasmodial slime mold, and it's called that because it looks like it's moving. If you watched it in time-lapse photography, it's moving really quickly and looks like it's growing and almost like an animal, like it's oozing. It has sort of an alternation of generations where it produces a stalk that's going to undergo, in the stalk, we're going to have meiosis, we're going to produce spores. Spores are going to germinate, form these two different types of cells, either amoeboid cells or flagellated cells, similar to male and female, but we don't call them that. They're both N, so haploid, and they're going to fuse to form a zygote, and that forms a new plasmodium. I think the cellular slime mold, these are usually brightly colored. The plasmodial slime mold, this is an example of a cellular slime mold. This is called dictyostelium. It's a very famous one because the spores form a packet at the top, and they literally, when it gets super hot, all the liquid, the fluid in this fruiting body got so warm that it projectiles the spores off. And this particular dictyostelium actually infects cow feces. So that's how people know so much about it. And this is called a cellular slime mold because notice it doesn't make this huge, it's more like an amoeba. It doesn't make that huge mass that you would be able to see like on a tree or on some leaves. This is microscopic, right? 200 micrometers. But they do have these amoebas that aggregate and then form these fruiting bodies. But again, 600 micrometers. You wouldn't be able to see this without a powerful, powerful microscope. Who else is in this group? The tubulinids, and the representative of that is an amoeba. So I'm looking for, I think he was way back at the beginning of our chapter. Go back and see, there we go. So this is a unicont. This is a free-living amoeba. It looks like Pelomyxa, which would be the one, like I said, we look at under the microscope. It has cytoplasmic streaming. So these pseudopodia they're called false feet because they extend out and they can trap things like, looks like it's going to grab this little algal cell here and eat it. If you remember all the way back or have the time to look back to chapter 7, I believe, at the very end of the chapter, when we talked about endocytosis, there was a picture of an amoeba that was actually eating a bacteria. The tubulinids, are all free-living amoebas. The entamoebas, which are a separate group, you'll see on your handout number 3, those are parasitic. There's only one that infects humans, and we're not its definitive host. We are an accidental host. It actually infects pigs. And then this last group here, again, if we look back to the original page, this pisciconts, notice this is including the nuclearids, the fungi, the choanoflagellates, and the animals. These last two, the two that are in yellow, the two that I've listed last on the handout, are the ones that are true protists, right? So let's look at the nuclearids first, because they are most closely related to the rest of the fungus, modern day fungus. They are free living unicellular amoeba that feed on bacteria and algae, right? And I wrote down there the closest living ancestor to fungus. If you want to read more about them, take a look at the beginning of the fungus chapter, and I think there's some pictures there as well. The choanoflagellates here, those are also free living, and they have a collar cell with some flagella, and they are the probable ancestors of, definitely of sponges, but notice they're most closely related to the rest of us, the rest of animals. I would remind you the importance of protists along with the fungus and bacteria. They're very important with nutrient cycling, which I think is what this picture is about, or it's a blight that's causing death. Let's put this picture up. Right, so they're super important for the food web because they're down at the bottom there, storing carbon, excess carbon that's in the environment, feeding everyone else, right? So we have the protozoan or the protistan producers. Those would be the ones that are photosynthetic. They're going to be ingested either by the heterotrophic protists and then larger plankton or larger herbivores or carnivores, which of course they're going to serve as food sources for larger fish, crustaceans, birds, right, and some mammals there. They're important for economic reasons. I listed there malaria, termites, the oak blight, that's what that was about. And again, probably the most important part that protists play in the world is they are this, with bacteria, they are going to be involved in nutrient cycling and serving as the bottom of the food web. Okay, so that ends the chapter 28.